I managed to put a presentation that I'm going to show you 3 of the technologies that we develop at my lab in the Oakland Engineering Institute. So, see, yeah, this works. So just to give you a little bit of context, usually when we talk about brain, what people refer to is the neural description of the brain. So neurons are even the minority of the cells that are in your brain. You have a lot of other cells like glia. So neurons are a big player, but there is also a lot of vascular control, and we can call vascular cells, involved in all the processes that sustain your cognition and your mental activity. So in my lab, we try to go through these 2 different worlds that are the vasculature and the neural brain, as one facilitates the activity on the other, but vice versa also we have a link that neurons affect the vasculature as well, So all the technologies that I'm going to show you are not developed by me by far. It's this group effort, and we are 3 researchers. I'm the research leader, but Finbar and Soroush are heavily involved in all of this work as well. We have 8 marvelous PhD students, 2 master students, 1 bachelor in engineering that is also gives us a lot of support and is helping us in developing the technologies. And of course, the guys that sustain this in a reasonable manner, that is our software team, that without them we would be completely lost and just only publishing papers, but with no coherence at all in our technologies. So the first technology is how we go from images to creating these digital humans. For the ones that you are not related with these words about brain modeling and what we refer with that in bioengineering, is basically have a computational simulation of how your brain performs, trying to capture as much as we can on the anatomy, but as well on the physiology for you specifically. And with this, we want to move for a more target clinical treatment of these patients and diagnosis as well, because now they will not be living with you as a point in a statistical distribution, but it's more treating you with all your unique features that you have as a human being. Okay? So how we do that? All the journey starts in Gisborne. So we take our images at the Matai Center. We have very specific sequences we run that allow us to see different kind of characteristics in your specific brain. And these are giving us some characteristics around anatomy, but sometimes also around physiology. And we are able to take all those informations. How we do that is with our in-house developed AI technologies. So basically, in this case, for instance, we have this image and we want to extract these vessels. Yep. So our AI technologies, in this case, it's a very famous one, it's called UNET, is extracting different kind of aspects of your brain. We call it features, and each time extracts more and more. And once have a large number of those, we'll go backwards and start combining them together to start elucidating what are the combination of these features that gives you the vessel. What you're going to see now is here. This is a probability of vessels, and we finally extract this and pretty much match what we want. Yeah. So once we do that, we can do these very nice things of recovering your specific brain anatomy, your vasculature. We can look at internal parts of your brain. For instance, in this case, it's the ventricular system. And here we are only showing a few and to make the animation nice, but we can extract all different kind of brain regions and also these marvelous white matter tracks. So what we have at the end is when we integrate all of this together, we get this kind of explosion diagram of the brain of a person, specifically this, you see the face, probably. This is myself, yeah? So, and this is my brain, in fact, so we scan it here in the So you can see the gray matter, the white matter, different kind of gray matter structures in the subcortical areas. You can see the ventricular system. You can see the vasculature here, a piece of the cerebellum. And why we want all of that? So makes a very cool image, but on top of that is that with knowing the anatomy, knowing the vessels, we can now simulate, for instance, how your blood perfuses your brain. Yeah. Here you have in blue what our model predicts, in red what is a real measurement. So we are quite happy with the precision of that. But you can argue, I can get this also from 4D flow MRI. Yep. That is a sequence that you run in the scanner. But the thing is, you have to get, if you do that, you have a given resolution and only you can see certain kind of measurements across the brain. With the models, we can have a continuous definition. That means that we can pick any point in your brain and know the velocity and the pressure. Yeah? And pressure is something that the scanner cannot get for you. The only way is just sticking a catheter inside your arteries in the brain, which can have several problems. So you don't want to do that in routine. But these methods allows you to have all that information non-invasively. So how we use this for diagnosis? We have currently 2 projects that has been supported by Maso and HRC. The first one is around how is having the brain perfusion for a specific person, and if we are able to retrieve all these what we call biomarkers that are basically physical measurements that we take around your body. In this specific case, what you can see in this cryptic diagram, that is what engineers we like, it's are the profiles of the blood flow. That means the velocities of the blood flowing through different parts of the vessel. And this is quite important because the velocity of the blood near the vessel wall is quite important for the development of plaque, for the healthy of the lining of your vasculature. So these simulations allow us to compute that across all the vessels in the brain up to 1 millimeter of diameter. Other aspect is also the pressure. Also, we can simulate the presence of a clot and how it travels through the arteries, and with that we can predict different outcomes if you have a stroke. So all of this can be quite informative for the practitioners. A second application, and this one has been quite successful so far funding-wise, we are developing with the sequence, the AMRI, probably most of you know it, is the one pioneered by Sam Holsworth. And we are collaborating to integrate this marvelous technique that allows us to see microdeformations in the brain to actually predict what is the pressure in all the other components of the brain that is related with this amount of deformation we see. Yeah. Why this is important? Because the only other manner of knowing the pressure inside your brain is putting a catheter by making a hole in your skull or by doing a lumbar puncture. Neither both are ideal if you want to do this in a large cohort of patients and non-invasively. So we are developing techniques to be fully non-invasive to make these measurements. And finally, this also fits back on research because now we can have a complete anatomical and physiological model of you and can be performed a lot of experiments that otherwise cannot be performed in a living person. Yeah. So things like making different stimulus on how the blood is propagating your brain, blocking one artery or putting more pressure in another artery and see how the blood distribution change. We have also a nice sandbox to try different biomarkers. Here we are developing one that is related with brain pulsatility, and this is blood flow pulsatility and how that can be a promoter of dementia is our working hypothesis. And what we have found so far is for a small number of patients that we tried this hypothesis, we see that the data seems to suggest 2 different trajectories with this biomarker, and this can be one potential biomarker for early detection of dementia. Other aspect, and this is one of the diagrams that probably are not very general public, is try to understand better how the different cells in the brain communicate. And for this, we want to specifically know how neurons communicate with astrocytes and with vessels. Because when you have activity in a neuron, that will provoke a cascade of events that will result in a vasodilation in a vessel, and that vessel will give more oxygen and nutrients to that specific neuron. And that is just a maintenance mechanism that you need to support your brain activity for longer. Yeah. So this is other dimension that we are now not using the simulations to create immediately clinical translatable biomarkers, but now we are using our simulations to try to go back to the science and understand how these different cells communicate and propose hypotheses. For instance, if we say that, sorry for that, if we suspect that the neurons and the astrocytes communicate through different channels or with different signal mechanisms, we can simulate that and see how that will match with experimental data or not. And then we can rework our hypothesis until we have more reasonable ones that at that point we want to spend more money and actually make experiments, be it in animals, in humans, or any other kind of trials. So yeah, pretty much that's me. So if you guys have any doubt or you have interest to know more about what we do in the group, please contact us. We are more happy to receive you, to give you a tour through API, and also to show you what other technologies we have at hand. And finally, sorry, I need to mention my funders, otherwise it's going to be a big problem. So I'm really grateful for the founders, for HRC, MBIE, Martin, Horizon Europe, that allow us to continue all this research. And obviously, I'm also grateful for all the marvelous collaborators we have. We have Matai and people, many familiar faces today from medical health science. But we also have international collaborators because all these models, all the physics behind this, all the mathematical methods, all the computational methods is something that requires a lot of know-how and a lot of talent. And this is our collaborations with MIT, Emolab in Brazil, Oxford, Max Planck Institute, and University of Trento, and recently, Pompeu Fabra in Barcelona, Ali Champy. So, thank you very much for your attention. Cheers.